animals have involved diverse anti-predator defense such mimicry, camouflage, voting colors, and like crypsis as well. So crypsis, mimicry, and voting colors have been extensively studied, but one of the deception, like masquerade, remains largely unexplored, particularly in spiders. So today, I'm just trying to share some stories about what we did on this topic. So from here, you can see the spiders I'm going to use, that's the system. And then I use chicks as predators. And also use other predators as well, like other birds and jumping spider like poacher, and also use flies as prey for testing ideas. And then also doing this visual modeling, and also use 3D printed spiders to test ideas as well. And in the future, I'm not trying to look at some chemicals used for this masquerading as well. And in future, I hope, doing this evolution of the masquerade in this group of spiders. So today, I'm just trying to look at the bird-dropping masquerade in this particular genus, Phrynarachne crab spiders. I'm not very good at pronounce the Latin species. So I just give overall about what we have done in the past 10 years. Before I go into those details, I just would like to thank the people contribute to the work. So the work's mostly done by the students, as a PhD students. And then lots of students also contribute as well. So you can see here, they're all jumping spider nymphs. So Daiqin is work on with in the last year, work on tracker. And then Bernata is work on Sina. And then Zoe is work on Sina and also other very beautiful species called Osema from Malaysia. And then Rachel's work on cosmophysis, mostly focused on opposite genes. And then students from high school even work on some jumping spiders genome stuff. And another student's work on anti-mimics. Okay, so the first one I have to thank Linda to invite me to come to here. That's my first time to attend AAS meeting. Thank you very much. And also thank you, Linda and Cole, to host me. And I stayed 2 nights in the beautiful house and beautiful forest houses here, gardens. Okay, so, and also lots of other students enjoy me working this well. So Neil from China, and the students from China as well. And then some students from this postdoc from New Zealand. Yep, so a lot of people contribute this work. What I'm going to talk today, I'm just give a briefing about camouflage and masquerade, and also give some briefing about this particular genus, Phrynarachne, and also most importantly, how to test the ideas about masquerade in this group of spiders. So I provide some empirical demonstration of this bird-dropping masquerading. And also looking for the dual function of this masquerading, and also looking for the mechanisms behind the this masquerade. So how this masquerade works. And I just gave some focus on this ontogenetical color changes during this particular species, and also looking for the ratio and location of dark light patches in this particular species. First, I just introduced some briefing about camouflage. Camouflage is very known, so it's a very common strategy of anti-predator defense. So the animals can avoid predation by protecting detection. So like there's background matching and disruptive colors. That's very famous. So I don't give more details. And also they can avoid predation by protecting recognition. So that's one example about mimicry. So also famous as well. There's some examples in our spiders and famous in butterflies. And then I'm introduce another one is also they can preventing recognition. That's the masquerade. That's what I'm going to talk today. So when animals resembles in inedible or uninteresting objects such as leaves and bird droppings or even stones. So this is some example. That's the butterfly. So that's the lizard, and then there's the insects, and then that's what I'm going to talk today, and some mantis. And bird dropping masquerade is very common in animals and particularly in spiders. So there's some examples about the bird droppings. I'm not going to focus on, I'm not going to talk on many others, but focus on this group of the crab spiders. And then I'll give some briefing and observation in nature. So that's all the species we collect by ourselves in the past few years. You can see the range of the colors, and there's huge variations in color and shape in the size. So that species distribute in particularly in Africa, East and Southeast Asia, and there are 35 species over the world. So there are 10 species in China and 12 species in Southeast Asia. So it's 22 species almost in Asia. And then Africa, 9 species, and Pacific Islands, about 4 species. And then the bird-dropping-like masquerade. So it's all often cited as a textbook examples, as natural selection. And then this looks like and also smell like bird dropping as well. And considerable variation in color, size, and shapes. So you can see here there's the many species we collect in China only, and multiple species, even in one spot, we collect 9 species over there. So it's a huge variation in dark and light patches, cross and within species. That's the one observation. And also you can see the body shape is different. So the body with or without tubercles. So that's one is a smooth, without much, without tubercles. Some with numerous large tubercles like here, very large one, and some with small non-tubercles, or sometimes with a numerous small one as well. And then there's a polymorphism in single species, within species. So you can see here, there's a one species in China. So you can see there's a color differences. And also you can see the female bias ontogenetical color shift in the single species. You can see we rear them from the first from eggs to adults. You can see there's a changes in colors. And then the adult females, the adult males is just small and dark. It's not bird-dropping-like. And then you can see in the field, we can look at some fly insects approaching the spiders as well. So that's what I observe in nature. And so there's a lot of patterns. And the question is here, why do this particular crab spiders look like bird-dropping? So specifically, we're interested about what's the function of bird-dropping resemblance in this Phrynarachne. And then what are the factors that influence the bird-dropping masquerading in this group of spiders? So we have observing the body size, changes in body size, and or maybe density, or maybe the dark light patch ratio, or maybe the presence and the size of the tubercles and ontological changes and color morphism, color polymorphism. So there may be other factors influence the efficacy of bird-dropping masquerading. And also, how has the bird-dropping masquerading involved in this group of spiders? So, but I'm trying to focus on these 2 questions. First one, I try to use some experiments to test ideas about this masquerading, bird-dropping masquerading. So provide this empirical demonstration of bird-dropping masquerade in this group of spiders. And so far, there's no empirical or experimental evidence to show such a bird-dropping masquerade in spiders, in this group of crab spiders. So just say in general, not in spiders. Just most assume there's a bird-dropping masquerade. So, but its problems, one, maybe the methodology is very challenging. It's hard to provide evidence to show this bird-dropping masquerading. So that's the big problem. So difficult to determine whether a predator has detected or just misidentify the prey, or maybe just simply fail to detect the prey items and then ignore them. So that's very difficult. But what you can do is just try to manipulate some predators' experience with the prey, and then you can test them. So that's how the John scalpel used as a particular caterpillar is a stem masquerade. That's the anythorn caterpillar. And then they try to use the chicks as a predator and then manipulate this particular stem and change this visual appearance against this stem, and then test the chicks with or without experience with this stem, and see whether they can learn from this training and then avoid this predation. So that's the chick they use. That's a domestic chick. So we try to use the same methods and then test the ideas or test there's a bird-dropping spider masquerading in that group of spiders. So that's the species are going to use. That's from collect from China and wider dispute in East and Southeast Asia. And sexual dimorphism in size and color. So that's the size. So that's a female, that's a male, so very tiny. And the colors are different at all. So, and we collect from Yunnan province in China. And then we just follow the same procedure as what they did for this caterpillar masquerading. And we train the chicks bought in China. So definitely we have a theoretical improvement with this. And then we train the spiders, we train this chicks, and then divided them to 3 groups. One was training with there's a bird dropping, and the other one is the bird dropping, manipulate bird dropping. So change the visual appearance. And then there's empty, means just a control. And that's the setup for this. I'm not going to give it more details here. So normally we have an experimental arena, and that's the body arena, provide some body chicks to provide the comfort to the testing chicks. So yeah, so mostly reduce the stress. And then we try to use this, collect this bird dropping from this Chinese bubble. And then once how we test them, we just test the chicks with this bird dropping and manipulate bird dropping, and then we just recorded what the latency to peck or attack this bird drops, and also look at the numbers of the pecks to this bird dropping or manipulate bird dropping. So you can see here, there's no much differences in the latency of the peck during the training season, training stage. And also there's no differences in the number of the packs. We did the 3 trials, 3 training, and then it shows there's no much differences in latency of the packing and the number of the packs. So it means they are willing to pack either bird dropping or manipulate bird dropping. So that's how we can train them and then get the experience. And then the next stage we try to do this predation experiments. So we divided chicks to 9 groups of 12 chickens. So that's, you can see here, one is 3 groups with experience with the bird dropping. The other one, 3 groups with experience with the manipulate bird dropping, and then control 3 groups as well. And then we test the different stimuli. One is the bird dropping spider, the other is the bird drops. And then the another spider is a normal, typical, long bird dropping crab spider. I don't give the names there, so it's just say it's different. It's not bird dropping look like. And then that's a testing. So we same setup, and then there's the chicks, and put this stimuli on this leaf, and then there's a provide 2 body chicks provide some comfort to the experimental chick. And then we recorded the latency of pack, means the time elapsed from start of the trial to make the first pack or first tag at test stimulus. And then see whether have some differences with the chicks with a different experience. So that's results we found that. So there's a significant differences in this latency to pack between the chicks with the previous experience with or without bird dropping, or with experience with the manipulate bird dropping. So you can see here, the chicks the chicks with the previous experience of bed dropping took much longer to peck or attack this bed dropping spiders. So BDS means the bed dropping spiders, and also the bed dropping as well, compared to the chicks with manipulate bed droppings or controls. So that means they had experience and then hesitate to attack the bed dropping spiders. So provide evidence the chicks may misidentify the bed dropping spiders as the bed drops. There's some videos that shows, like say there's a chicks already see there's a bed dropping spider, they just ignore them or hesitate to tag. But the next one, if you provide this, provide this non-bed dropping spider, the normal typical crab spiders, and then the chick doesn't work. And then the pig and then eaten, okay? So in this experiments, we provide this, the evidence to show that this bed dropping spider may form bed dropping masquerade that function to promote misidentification of the masquerading organisms. And also this experiments shows the predator's collective restricted, including this recognition and identification, rather than the sensitivity or capabilities, and the selective force driving the evolution of the bed dropping masquerade for the first experiment. And then there are different types of masquerade. One is called this element imitation. So that's the organisms resemble some specific and common object that is one element of the common environment, like the leaf or stem or some things like this. And then the other one we call object imitation. So the animals resemble some specific object. It is not common and not elements of the environment, like bed dropping and stones. So there's two types. And then from this definition, we can see some differences between these two types of masquerade. So the element imitators are viewed against background. So that including the samples of the type of item, means the object, that masquerade, they masquerade us. And the object imitators are reviewed in isolation. So it's not necessary to be there in the environment. So that's some differences between these two types of the masquerade. So if that's true, and then we may see there's element imitators and their models are viewed simultaneously by a predator. And in this case, they have to compare to this two between like this object and this masquerader. So that's and the very stronger selection to resemble their models than object imitators. Means if you're the imitators like bed dropping spiders, the bed dropping is not necessarily be there, so they don't need to compare both of them. So they use this brain or template to recognize whether it's a bed dropping or not. So in this case, we can predict that element imitators may have to be closer or more or less perfect mimickers than object imitators. So in this way, we can predict that Phrynarachne spiders can benefit from bed dropping masquerade as object imitators, regardless whether a bed dropping model is present or not. So that's our predictions based on this types of the masquerade. And then we did same experiments, use the chicks as predators and training them the same way as what we did. But in this case, because we try to look at whether the presence of the bed drop or not will affect the efficacy of the masquerade or not. So that's the training, the same. And then we tested, it's different from the first experiment. We use two stimulus. So stimuli here, there's the three groups here, the chicks with previous experience with the bed droppings, and the others manipulate experience, and then there's a control. And then we tested two bed dropping spiders or two bed drops or one bed dropping spider and one bed drops. So that's how we can try to look at the differences between this one and this one. So see any differences if the backdropping is present or not present. And then the same setup. And then we try to look at the similar parameter, the latency of the pack. And then that's the results we found. And then you can see from the first experiments we did use a single stimulus, only one backdropping or backdropping spider or a manipulate backdropping. And here we have two backdroppings or two backdroppers or two backdropping spiders or one backdropping spider and one dropping. So that's the results, quite similar as this one with the single stimulus. So means even you provide the backdropping with this backdropping spider, and then they still provide this provide evidence to show this masquerading, the backdropping masquerading may promote misidentifications. Okay, so we give it more details so you can see here. There's no much differences between these two. Either you have one stimuli or two stimuli, and then the chicks training with the backdropping took longer to pack the test stimuli than those training with the manipulate backdropping or naive ones. So that's one suggested the chicks misidentified spiders as backdroppings. And then you can look at the other thing is that's the empty ones. So they are much smaller. So it's take short time as well. And the control is that one I didn't mention about this non-typical. There's a typical non-backdropping look like spiders. So they always take a short time to pack up or attack them. So they eat them easily and in a short time. And also the chicks gave only spiders. So either it's only one spider, backdropping spider or two backdropping spider, took a similar time to pack the stimuli as those given only backdroppings as well. Means they have a similar functions there. So they consider, the chicks consider this backdropping spider as a backdropping. So that's some results. I did provide this stats. And then also you look at the backdroppings as well. So that's quite similar in the latency of packing if provide a single stimulus or double stimulus. And the most important one now we can find out. So the chicks training with backdropping took a similar time to pack the spiders when the backdropping was absent, like here. There's only the backdropping spiders, but that's why you can see here when the chicks was one backdropping and one backdropping spiders, and then they take the similar time to attack or the stimulus. So this experiment, this result shows the spider benefit from the masquerade regardless of whether the backdropping is present or not. So that's the results you can see there. And then from here, we can see these spiders are likely to misidentify as backdropping, whether when backdropping are present or not. And thus the function as an object imitator. Now, the first two experiments provide empirical demonstration of backdropping masquerading. And then because the spiders are predators, definitely they need to catch the prey. So that's how we see whether they have such a function as well. We come with this a dual function. First one we call the protective masquerade. The other one we call aggressive masquerade. So what is the difference between this aggressive mimicry and aggressive masquerade? The aggressive mimicry, like jumping spider, like Portia, they mimic Deborahs and then catch the web-building spiders. And then there's a mimic this Deborah and invade the webs and catch the prey. So that's what we call aggressive mimicry. When the predators mimic harmless or benefit stimuli, the stimuli can be visually, chemically, or even acoustically similar to an evil object to lure and capture or otherwise exploit their prey. But this one, the aggressive masquerade, is the masquerading predators resemble this inedible and uninterested objects like leaves or backdropping. So that the prey species can detect masquerade, they can detect the masquerading predators, but the misidentify them as the innocuous items, are not interested, cannot eat. So make them less wary and easy for the predators to catch them. So that's what we call aggressive masquerade. And then how the crab spiders to get access to prey in the field. So that's why we do some field experiments and test this aggressive masquerading ideas. So we predict the species, the spiders may aggressively masquerade as bed dropping in order to catch a potential prey. So we did experiments in the field in Yunnan province in the tropical forest. And then there's some plants here. We found many individuals in this particular plants on the leaf, on top of the leaf. So there's the spiders. And then there's the eye stalk flies. It's easy to find them there. Okay, so we use the system and try to test this masquerading, aggressive masquerade. And then we first want to do some field surveys and some differences in prey catching in bed dropping spider and bed droppings and control. So that's how we just locate the spiders in this leaf and also locate some bed dropping on leaf. And then we use some empty leaf as control. And then we find out in both bed dropping and spiders, they attract a lot of the dipteras and similar in the prey types, but some differences for the control. And most importantly, we look at the capture rates per hour. So how many individuals of prey they capture per hour? We use a video recording and playback and recording how many individuals captured by bed dropping spiders and bed drops and control as well. So that you can see here, both bed dropping spiders and the bed dropping capture a lot of the insects. And then that's the type of insects. There are not much differences between this bed dropping spider and bed droppings. Even the higher in a higher number attraction the rates than in the bed droppings than spiders. Maybe the bed dropping have a much strong chemicals. We don't know yet. I'll have to test it. So at least here we can see both spiders and bed drops attract species insects at a significantly higher rate than the control leaves. And then to further confirm this aggressive masquerading, we also did some manipulated experiments to paint the spiders. So we have a non-manipulated spider, and also do paint and shame. So it's some chemicals because we did paint to dark like this, blackened and then whitened. So there's 4 groups of spiders and put in a single leaf, and then we use a video recording to look at what's the insects and how many insects attract to this different types of the groups of spiders. And then we found out, and then there's a significant differences here. So you can see both manipulated and shame spiders attract more insects compared to this blackened and whitened. Even the whitened is a little bit higher than this blackened one. So that's how you can see some differences, but there's not much differences between in the prey attraction in between bed dropping and bed dropping spiders. And if you look at the groups or types of the prey, there's not much difference among them. Okay, so from here we can see a variety of the dipterans and hymenopterans were able to detect this crab spider, but are still lured by them in large numbers. So also we can see that insects lured by the spider may misidentify the spiders as bed droppings. So, and the bed dropping masquerading may serve as aggressive masquerading in addition to the protective masquerading in that species. And now I move that once we have this functions about why do the crab spiders like bed droppings, and then according to our observations, lots of the patterns are observed, like the size, and then the white and dark patch, the ratio or proportion are different among species or within the same species. And also there are many other shapes as well. It's different. So that's why we try to look at the factors, including the efficacy of the bed dropping masquerading, focus on two experiments. One is the ontogenetical color change. The other one is the ratio and location of the dark and light patches. So I'm not going to give much details on the second one because of time. Okay, so from our observation, we did a life history of particular species, not just that species, many other species as well. We use this as example about looking for the ontogenetical color change. Why ontogenic changes? So you can see here, that's the pattern. Our question is, say, why color-based defense shift from crypsis to masquerade? So assume maybe this is driven by differential changes in predation risk of two defense strategies with increasing body size due to ontogeneity. So look at this one. We just try to do some measurements of the colors. You can see the changes of the colors from the fourth instar here. So the second and third instar are black. So the whole body is black. And then from the fourth instar, you look at the first leg femur. They have some lighter or brighter colors here. So some differences here. So you can see here the increase in the brightness of the first leg femur. So here, that's some increase in the changes in the colors. But question is why have such changes? So we use this 3D printer model spider and try to use the chick as the predator, but we don't train them this time. We just test them and see how they respond to this different colors with a different size and different development stages. And then we can see the different predators like the chicks cannot distinguish between the live spider and the 3D printed spiders. Okay, now allow us to test our ideas, use the chicks and use this 3D print spider. And then that's the 4 types of the prey. And one is a dark model, it's small and large. And then the other one is a bird-dropping-like model, it's small and large. The same size, like here, you can see the small and small, the same size, but the color is different. The large one also is the visual experience different, but the size is the same. And we use the same testing setup, but no training. And then we record the probability of the attack, yes or no. And then we also look at the detection time, means the time elapsed from the start of the trial to the test chick that oriented and started approaching the model spider. And then also once they detect it, and then whether they're going to attack the stimuli or not. So we record a hesitation time. So the time taken by the testing chick attacking the model spider after detection. So the detection means they can detect it, but they may not to go into attack because they're very cautious. So that's how they may take longer time to attack the bird-dropping-like spiders. And then that's our results. It shows here. There's a small black, and there's large black, and there's the small bird-dropping-like. That's the large bird-dropping-like. So you can see here, that's the percentage of the spider attack. And then you can see here, there's a pre-color and size along, and no significant effect. So if you compare to this one, the small altogether and this one, there's no much differences. But we found a significant interaction effects between pre-color and size. So we can show here the large spider was attacked more often, but only when they are dark, but not bird droppings. You can see here. Yeah. So that's the results we can tell the ontogenetical changes in color because of the predation risk changes due to the body size. And also look at the detection time and hazard time, and also show some differences here, quite similar as the probability of detecting. So if you are small, and then the spiders take longer time to detect them. And if you're bright, like some bright colors in the first legs, and then they're easy to detect them. So this reasonably, right? And then if you look at the hazard time, you can see here, if they are dark, regardless of small and large, they have no hazard. They just try to attack them. But when the spiders are looks like a bird dropping, they take like longer time to attack. Okay, So from here, you can see the increasing predation pressure on larger and the cryptical spiders during the ontogenetic. So, and then switch to backdropping masquerading may be more effective as they increase in size. So ontogenetical shift from crypsis to masquerading is not adaptive in that species, and thus spiders may gain the protection from masquerading. So that experiment all shows or provide evidence that's masquerading, backdropping masquerading in that species. And then the other factors. So other factors, we try to look at I see there are considerable variation in the proportion and location of the dark and bright patches, cross and within species. You can see this, there's a brighter, there's a dark. And then you can see the same species, they may have some very, the range in the dark patch and white patch. And I see the same species as well, the other species, the different species, and then the proportion and the location of the dark and bright patches varies as well. So why such differences cross species and within the same species? So we just look at this one and try to see whether the efficacy of the backdropping masquerading in that species. We use the same species, different species, but we just try to come with some model that range of the dark and white patch percentage or proportion from 0 to 100 here. So it's all black or 25% white or 50% white or 75 white or all white. And then there's a control spider, it's a long backdropping spider as a control. And then we also try to look at the model spider with the different white patch locations. So same percentage of the black, dark and white patches, but varies in the locations. Same for this 50% white and 75 white. And then also look at for the model spider as well, small, large in different groups. And then this time we don't use the chicks, we use a Porsche as predator. Okay, so no training. And that's the setup. So the Porsche as a predator and put here, and then whether they can cross this threshold and look at attack this 3D models. We use the 3D print models. And then we try to measure the detection time, attack latency, and also proportional model attacked it. So that's some experimental design. Look at the effects of dark light patch proportions. So that's the model spiders with a different proportion of white. And then you can see here this proportion of the model crab spider attack. And then you can see there's no much differences among this. So from all black to all white, there's no much differences, no significant differences. But that's why it's easy for them to attack. So it's 27 out of the 30 spider attacked. And so the predation attack a backdropping-like and all black spider models less frequently. And then no significant differences in predation among spider models with this different white and black patches. And the all white spider model was attacked more often, a little bit more often, but it's not significant. And if you look at the attacking time and attacking latency, so detecting time, you can see here, there's no much difference from all black to white, but this one is much, much shorter for the control spider. And then if you look at attack latency, and also there's significant differences between this all black until 75 with this all black. So the all white one, spiders attack in a short time, and similar as this control spider. So the predators took longer to detect and attack a backdropping-like and all black spiders. And then the predators attack all white and control spiders much earlier. And then look at the locations. So there's a 3 different proportion of dark and bright patches, but very different body parts. And then we test it, and then that's the results, the percentage of the model crab spider attack, and then you can see there's no much differences. And you look at the detection time, and also attack latency, except there's a 50%, some differences here, and then there's no difference in other groups, according to the regardless of location of the black and dark patches. And about size, so the spider size and corresponding dark light patch proportion had no significant effects on this likelihood of attack, except the control spider. So that's the non-building spider, non-backdropping spiders. So from here, tackling time also no much differences. So from that experiment, you can see the proportion and location of dark and white patches, and the body size of backdropping-like model spider are little or no significant effects on this efficacy of backdropping masquerading in this group, but it's the predators, not chicks, is the portia. You can see some difference in between this small dark and the large dark and small backdropping and backdropping. So the portia cannot detect differences. So the variation, the various visual appearance, body size, and different white dark proportion and location may have been involved by this Phrynarachne species because they confer similar protection from that experiments only. So overall, we tested a form. So what are forms of this backdropping spider across the species, racing species, and also the function of the backdropping masquerade. So we found that's a dual function, particularly protective and also aggressive masquerade. And also we found this ontogenic shift in defense strategies because of the size, the body size change, the color change, and then the defense strategies also change as well. And then we also look at a few the factors which can influence the efficacy of the backdropping masquerade. So that's the mechanism of backdropping masquerade. And the last one, this study shed light on our understanding of the function and evolution of backdropping masquerading in animals, particularly in spiders and particularly in the crab spiders genus. But as I mentioned, so lots of the observed patterns we found in within species, cross species. So we are still testing and doing some experiments. So that's ongoing and future studies. So the mechanisms of backdropping masquerade. So size, because the backdropping varied in size, whether the size of a backdropping, the chicks or predators training with a different size of backdropping, the backdroppings will affect the efficacy of masquerading or not. And also the density of the model objects means how many backdropping spiders and how many backdroppings in the environment or affect this masquerading or not. And also the view distance and orientations as well. And other predators such as birds and lizards can be used as predators in the future study as well. And also we are very interested about evolution of backdropping masquerade. So the origin of this masquerading and the evolution in this particular genus. So you can see here the polymorphism. Why polymorphism in this particular species, in many species actually, but this one is quite obvious. And then, so we can also look at the size of backdropping. So we can train in the chicks with a different size of backdrop and here, and then test them. And also the density of the backdroppings as well. So we can train in the chicks and then test them in different density or ratio of the backdropping spiders and backdropping. And then also looking for the shape of spiders, as mentioned, they have some is a smooth, it's no typicals, some are small typical, some have large ones. So why such a difference in shapes? So we're trying to use this as 3D print models and test ideas and training the chick and test them as well. So that's all about it. Thank you very, very much. Yeah, any questions? The white coloring is one. We don't know, so we haven't looked at that yet. So I'm wondering if they could actually reinforce themselves that they're full of wanting and the bird doesn't. Yeah. A don't get the first group. Yeah. But if you do. Yes, we did some GCMS and GCEAD test, and then we found that the adults and juvenile, adult female and juveniles have the same smell as well. And also we found a particular peak which are already identified when I was trying to use the fly to test them whether the compounds is response to this or not, or check the flies, but not confirmed yet. We just do a few trials, and also because we are interested in the evolution as well. So whether the same compounds cross the same different species or different life stage as well. Yeah.
Don't the chicks from the previous experiments, the earlier ones we talked about, don't the chicks get presented with their own droppings?
Yeah, so it's a good question. So they may, but so when I didn't give the details about how we're training, how they yes, and also we put some papers, and once they drop, and just to remove it to clean them. So try to avoid such a thing.
But maybe it starts to test your idea about the size, because presumably the bird dropping, the model they get is bigger than their own field.
Yes, that's why we're going to try to test about it. Yeah, exactly. Definitely. So we observe the back dropping in different sizes. But some is very huge ones.
Right.
Yeah.
It really stuck out to me when you talked about like they don't only look like bird droppings, but they smell like it. Is that purely subjective? Like, you feel on the tire, so yeah, that smells like bird dropping. Or did you do like, is there some sort of chemical analysis looking at like the chemical signal composition from the bird dropping versus spiders?
Yeah, I understand your concerns. So we try to do this at GCMS with bird dropping spider and also bird droppings from different the bird dropping collect from different birds as well. We try to use a GCMS and see whether they have some similar compounds. And then we also use the GCEAD to test which compounds may have such a biological meaning from bird dropping and bird dropping spider. We try to control this one. So, but it's different. It's very difficult to control. But first, I have to see what the compounds, which may smell like bird dropping and attract some insects or may repel the predator. Yeah, I have the results from repelling and prey attraction. Use a housefly as a prey and poacher as a predators. We use the chemical cues, also the visual cues, along and both together. It's a huge experiment result. I have no time to present, so I deleted from there. So, yeah. Thank you. I haven't get your question yet. Or in the lab. So not much. So, but also we don't breed them for more generation, just one generation only. So try to avoid some changes in that. Yep. Thank you. You have some question? Yeah.
I was just curious about like the shapes on like the spider and like how bumpy they are and things. And if that's like related to kind of predators that live in the environment. So like if the bird ate like one fruit, leaves, like flies.
Yeah. Very interesting question. So that's why I'm interested. And then we're testing at the moment. So because you can observe the species with these patterns, and then and the pool also some small, some like some tubercles as well. So just try to train the chicks with different types of or shape of the back dropping and then test it. And also we're interested about the evolution of this one. So start with a smooth one until the big one or not. So we try try to come up with phylogeny and map this patterns to the tree and see how. Yep.
There are species within this genus that do not mimic bird dropping.
All species are, yeah, all right. 35 species based on that description. And many species just described in 19, very early from Africa, and we don't know exactly. So that's why I'm very interested in to look at the species in Africa. We planned the field trip in Africa, but the COVID starts, so couldn't make it. Yeah, you.
That's amazing, thank you. Something I know, if they choose. That's the bottom one. Some reason we in a particular place.
Yeah, that's very interesting question.
Repeat the questions.
Oh, okay. So whether the bed dropping spiders sit in a particular environment or microenvironment, right?
Yeah, and
Choose particular size close to the bed dropping or not?
there has to be a bird nest to
Oh, okay.
concentration.
Yep.
Colonial.
At least in the place we work there in the tropical rainforest, it's understory. There's not much the nest over there. So mostly just some birds that's flying and then make a drop in. And then we see a lot of the bed dropping on the leaf, but it's randomly. It's not particular, but also spiders particularly just sit or choose the leaves with more bed dropping or not. So it's random. Yeah.
I noticed that there's often sitting on little silken pads.
Yep. Yep.
Do you think that they can adjust their white and dark proportions on the fly quickly by adding more or less silk?
Okay. So very interesting questions, and also we observe in the field definitely, and also in the photos as well. And we try to look at this one, but the sample size is not big. So that's why we didn't present field work. So we use this one as consider as a factor, presence of silk or without silk, whether affect the prey attraction or predator detection or not. So the sample size is very small. So that's why we didn't present this one. That's very interesting question. And also because the silk also, we test whether it has some bed dropping smell. So we test with flies and Porsche as well. The Porsche avoid the silk, and then the flies attract to the silk as well. Yeah. Interesting question. Very interesting question. Yeah. Cool.
But they're also observing this testing. I wonder if this 1650, is there a test those to see if they're linked to a shorter after watching?
Wow. Okay, we haven't. Okay, so Cole's questions about whether the body chicks the presence of body chicks will affect their behavior of the testing. We didn't observe this one, and then also we didn't analyze the data. Honestly, no. Maybe I'll go back and play back and look at the behavior of this. Very good, very good. Yeah.
Were buddy chicks ever used as test subjects?
No, never. So we just separate them, and even we made put in a box and separate with the testing chicks. And then we didn't give the details. We changed the body chicks every 2 trials. Yeah, the timing, sorry. Thank you very much.